after today's session, we'll feel good about it. Okay, so let's get started. And if people join, we'll just catch them up. But for today's agenda, I have a few activities planned. So we're gonna do riddles for the icebreaker, and then a Kahoot. I planned a really long Kahoot. I finished it yesterday, and I literally went through all the lectures and I just picked like the best questions that I could find from each lecture. And I feel like Kahoot's like a good format because, you know, it's like a quiz type of thing. And then we're going to review like vocab terms, like important things that we should know for the midterm. And then if we have time, because the Kahoot's really long, so if we have time, we'll go into concept bubbles and I'll explain what those are. And then we'll close out the session. Okay, so icebreaker riddles. I am so excited for these. I made these like from scratch. Like I put like my rhymes together and I hope you guys enjoy them, but you're gonna guess what these are, okay? So let's start. I have checkpoints you must pass. If you are cancerous, you will go through me fast. What am I? So please type your guesses in the chat, what you think this is. It's okay if you don't know. Think about what has checkpoints. What is like something that we learned this this exam block that has like checkpoints, like stages? And if it's cancerous, like it affects the length of the cycle. Any guesses? Mitosis? Good guess. Mitosis is part of it, actually. So the general thing, yeah, Rachel, yeah, you got it. Cell cycle. It is cell cycle. So cell cycle has checkpoints, it has stages, G1, S, G2, and G0 if needed, and then mitosis. So this is cell cycle. Okay, next riddle. Helping out in the replication fork has my heart. My main role is to keep the single DNA strands apart. What am I? So think like an enzyme that does this in the replication fork. Helicase. Good guess. Any more guesses? Chromatids. Good guess. Not quite, but these are good guesses. Think about what enzyme has the role to keep the strands apart from each other. I feel like you guys are getting closer. Single-stranded binding proteins. Yay, yes, you got it. Good job. Good job, all of you, for participating. But yes, single-stranded binding proteins keep the DNA strands apart after helicase has unwinded them. So that's the difference between helicase and SSB. Okay, let's move on. Against viruses, I shall rebel. My closest friend is the B cell. What am I? I love this rhyme. Maybe think the immunity chapter. Any guesses? I see a guess for antibodies. Do we have any more guesses? Same guess. Would love to hear what you all think. Frontline defense. That's a good guess. Yeah. Anything else before I reveal the answer? Okay, so actually, yeah, Rachel, you got it again. Antibodies, antibodies rebel against viruses because they, you know, kickstart the immune response. And the reason why like the closest friend is the B cells because B cells produce antibodies. So yeah, good job. Okay, let's move on. I am responsible for the pandemic. Because of me, things are quite hectic. What am I? Sorry, I love these rhymes so much. I feel like you guys, this one's like a little bit like fun and easy. Virus. Do you guys agree with that or any other guesses? Oh, sorry. Okay, yeah. Christian, good job. You got it. Virus. Yeah. Coronavirus is responsible for the pandemic and things are quite hectic because of it. this is the last one. Larger surface area helps me out a lot. People who like ATP think I'm really hot. What am I? Think like, surface area, larger surface area, ATP. What thing could that be? Okay, I'll just reveal the answer so that we don't take too much time. So this one's actually, yeah, it's totally fine. This one's actually mitochondria. So, mitochondria is the powerhouse of the cell. Mitochondria produces ATP. So that was like the clue, like people who like ATP. And also mitochondria has like inner membranes that like they look like that, like little squiggly lines in the mitochondria. And those are inner membranes. And the reason why there are these like inner membranes is because the purpose of it is to increase the surface area for more biochemical reactions to take place in the mitochondria. So larger surface area, like mitochondria has evolved to have a larger surface area for that reason, for the just a larger space to carry out these biochemical reactions. So yeah, hope you all enjoyed that activity. Okay, so now let's get started with the Kahoot. I'm gonna go to the Kahoot and just have your device. Okay, so, okay, none of us got this. It's okay, let's review it. So, oh, I didn't upload a picture for this. So, the cell cycle, there are 4 stages: G1, S, G2, and mitosis. And G1, you can think of G as growth. So during G1, the cell is going to be growing, just, preparing for division. And S is where the DNA gets synthesized. So the S stage is where the DNA gets replicated and like duplicated. So you can remember, a really good way to remember this is G equals growth, and then S is synthesis. So make sure, like, put that in your notes, whatever you need to do. G is growth and S is synthesis. So G1, the cell is growing. S, it like duplicates and divides. And then G2, or no, it doesn't divide, it just duplicates its chromosomes. And then G2 is just more growth and just directly preparing for mitosis. So yeah, hopefully we're good with that, but we will review this again later in the session. Okay. So yeah, I'm glad that, like most of us got this, but ribozymes, by the word ribosomes and enzymes, they're basically just like RNA that function like proteins. And so, the word kind of has like a protein and an like enzyme in it. So just remember that ribozymes are RNA that function like proteins. Yeah, it wouldn't be ribosomes with mutations or anything like that. So just a definition word. Okay, so this is kind of like not a trick, but the correct answer is all of them. So let's go over why it would be all of them. So gut cells, our gut is always going to be regenerated. So those are always going to be, dividing. So are skin cells. We lose like, I think, millions of skin cells a day. So think about it. If we're losing a lot, we're going to need a lot. We're going to need to divide a lot of our skin cells to make more. Embryonic cells, these are like stem cells. Stem cells divide like, crazy because they're undifferentiated. They want to get to where they're going. So embryonic stem cells would divide a lot, and so will cancer cells. Cancer cells are very aggressive growth. They go through the cell cycle uncontrollably. So all of these would divide often. Some cells that wouldn't divide often are like neural cells, brain cells, stuff like that. So yeah, let's move on. And look at the visual for this one. This yellow thing is the cell. Think about it like that. Okay, so let's review this. So increasing the NHE transporters means that we're going to have a lot of H plus leaving, because you see the H plus, how the arrow is going out. So if we have NHE transporters, H plus is going to be leaving the cell a lot, and a lot of Na is going to be coming here. But here we're not concerned about Na because we're talking about like pH, acidity, basicity. And when we're talking about acids or bases, we're thinking of like H plus because that determines the pH. And by the way, pHi just means like the inside of the cell. So we're looking at the yellow here. If we have a lot of these NHE transporters, that means that a lot of H plus is going to be leaving the cell, right? Because see the arrow, it's going to be leaving. So what happens when so much of these H plus protons leave the cell? What happens to the inside of the cell? Is it going to be more acidic or more basic if we have a lack of protons, a lack of H plus? It's going to be more basic. So basic equals increased pHi, pHi. So that's why these two would be like the same answer, and these two would be the same answer. But in this case, we're going to have a more basic interior. And if there's any questions, just put them in the chat. But good job so far. And I purposely like wanted to make these questions hard, so sorry if they're like a bit hard, but it's meant to be like that. Okay, so Miller's experiment, remember when he tested like the primitive earth. So he put like methane, he put water, he put ammonia, he put yeah, those are just some of the things that he put in. And what he found at the end of the experiment was that even with like no oxygen, like abiotic factors, amino acids were still produced. So what this tells us is that amino acids can be formed abiotically, and abiotically means like no life. So no oxygen was present or anything like that. Yeah, Christian in the lead. Okay, so panspermia, I'm pretty like we did learn about this, so maybe like you guys just forgot, but panspermia is just a theory stating that life originated from outer space, like bacteria came from outer space, and that's how we have life. So yeah, all of the rest are just like BS answers. They're not like valid. It's panspermia stating that life came from outer space bacteria. So write this in your notes. I'd encourage you to just maybe take notes of these because it might show up on the test, but yeah. So actually the red one, RNA created life, this is a different theory. This is called RNA world theory. Panspermia is different. Okay, so Yeah, so gene flow is the correct answer. So genetic drift would be different than gene flow. So genetic drift is a different situation. Genetic drift, you can think of like D drift equals disaster. So this is like a catastrophic random event that happens that like kills off a population. Gene flow, you can think about it like literally like the word gene flow, like genes are getting flown. They're like the regions are like they're migrating, and then the new region gets introduced to the gene from the other region. So like if a bird flies over to another island and breeds with another bird, that is gene flow. So yeah. Okay, I'm glad to see that most of us got this right. So genetic drift, like I said in the last question, the D, I would think of it as disaster. So in this example, like a person stepped on the ant population. This is like a catastrophe, it's like a random event. So this would be genetic drift. Genetic shift, there's no such thing as genetic shift. So that was just there to like trick you. There is antigenic shift, but that's different than, it's not genetic shift. So yeah. And it wouldn't be mutation either. Mutation happens like biologically. Okay, yeah, helicase. Wait, hold on, I think I have a chat. Oh, yeah, that's a good way to remember. Yeah, helicase. So not ligase. I know they sound similar. Helicase unwinds the DNA. Ligase is a glue. Just think of it like that. And I don't want to like give it away because I think there is a question about ligase, but yay. Okay, I'm glad that most of us got this right. So RNA rolled hypothesis. It's really easy to remember because RNA is like it's like in the word. It's basically just saying that life originated from RNA. And it's not like a fact, it's just a hypothesis. See you, Christian. Okay, so DNA is not the enzyme that's providing the starting point for the replication point, for the replication fork. It's not DNA polymerase. DNA polymerase synthesizes the new DNA strand, but it's primase. So I'm going to put it in the chat. Primase provides starting points because it releases the RNA primer, which indicate like where we're going to start. So primase provides a starting point, not DNA polymerase. Okay, yeah, I'm glad that most of you got this right. So the lagging strand is originally presented in 5' to 3'. So if it's originally from 5 to 3, then the new strand is going to be from 3 to 5. So to tackle this question, just think, okay, what is the lagging strand originally in? And you're going to think, okay, it's originally in 5' to 3'. So the new mRNA strand will be the opposite. And of course, if there's any questions about this, just put it in the chat. Okay, so the most important thing to note here is that DNA replication is not a steady speed. It is highly variable. Sometimes it'll be fast, sometimes it'll slow down. So it's not steady. It's very like changes. Oh my gosh, yay, all of you got this right. Okay, I don't even need to explain it then. Good job. Halfway through. Okay, I'm glad that most of us got this right. So yeah, proper disposal of batteries is important because if the metal gets into our bloodstream, it's going to affect our DNA polymerase and it's going to make it more prone to mutations, to errors. We don't want that. So yeah, it's all about the DNA polymerase activity. Nice scores. Okay, pay attention to the whole sentence. That's my advice. Okay, so okay, the reason why it would be false, ligase does attach fragments, that is true, but what is it attaching fragments of? It's not the leading strand, because remember the leading strand is one continuous strand. We are talking about, when we refer to ligase, we're talking about strictly the lagging strand. Yes, Rachel, yes, lagging Okazaki strand. Okazaki fragments are only found in the lagging strand, not the leading strand. So ligase is not relevant when we're talking about the leading strand. It's only relevant when we're talking about the lagging strand. That's why this would be false. Yay! Yeah, diploid. Okay, so yeah, innate immunity is going to have a way faster response than our adaptive immunity because remember, innate immunity is the immunity that we are born with. It is our first responder. It is always there for us. Adaptive immunity is also there for us, but the response is going to take a little bit longer because it's not like our inborn immunity, if that makes sense. So innate is always faster than adaptive. Okay, so yeah, it would be one light, one heavy, and let's take a look at why. So these are the binding sites, and it's asking what are the chains. So the blue The blue would be light because see here it's pointing to light. And then what is the red? Well, the red is heavy. See here it says heavy chains. So when you mix the blue and the red, it's one light, one heavy. Same with here. The purple is light, and then this baby pink is like a heavy chain. So each binding site is going to be composed of one light chain, one heavy chain. When these chains mix, then the antigen molecule is able to bind. Hopefully that makes sense. Ooh, nice answer streak, Rachel. Okay, so as the DNA polymerase ages, gets older, it's gonna decrease the activity as well as likelihood of accuracy. So as the DNA polymerase ages, it's gonna not only decrease its activity, but it's also going to be more likely to mutations and errors. And this is easy to remember because you can just think of like any organism on Earth. As they age, they're going to be less functional. Same thing with DNA polymerase. So as it ages, it's going to have decreased activity. The x-axis is days. This right here. Okay, I'm glad that we all got this right, but I think some people didn't answer. Oh, if there's anyone new that joined, feel free to join the pin right here. But yeah, you guys got it. Just look at the peak, and that would be the answer. Okay, I'm glad that we got this. So I get why maybe one of you picked 15, but let's just take a look at this. So 10 is going to be approximately right here. That looks like the peak. If we do 15, that would be in the middle. That's not really the peak. It's like actually declining. So for this type of question, just do what you need to do. Get a ruler, like measure it out and see where the peak is, and that would be the maximum. Oh my gosh, Rachel, 9 correct answers. Wow. Okay, so we have an even distribution here. So yeah, the correct answer, first of all, it's important to get vaccinated because of herd immunity. We're currently experiencing this right now with the COVID vaccine. I think as of a couple of weeks ago, we had 10% of adults vaccinated, which is really good. So this is herd immunity. So we are trying to achieve herd immunity right now in the US. Herd immunity is like trying to get everyone on the same page, trying to get everyone vaccinated, because this is how we got rid of chicken pox. This is how we got rid of measles. It's because of herd immunity. And also the kind of the molecular description of why we should get vaccinated is because getting vaccinated is basically you're putting the virus into yourself in a safe way, but this is going to trigger your primary response. So now when you encounter I'll just say COVID, when you encounter COVID for the second time, you're already going to have memory B cells ready. And this way, like getting vaccinated is important because you'll already have those memory B cells, and now your secondary response will be triggered when you encounter the pathogen rather than a primary response. So yeah, not to make the primary response faster. It's so you can have the secondary response ready to go. Okay, so antigenic shift and antigenic drift sound very similar, but they are different. So let's talk about what antigenic shift is. So a lot of you picked this. Antigenic shift is basically referring to, I'll give the example of the swine flu, the pig flu. So the swine flu was able to infect us. Basically, how is because a virus from a pig mixed with a virus from a human, and then those two mixed and then created a new virus. That's what shift is. That's what antigenic shift is. Antigenic drift is basically just referring to a virus that's accumulating mutations over time. And we see this with the flu virus every year. It's changing, like it's constantly changing. This is why we have new flu vaccines every year. So antigenic drift is basically just referring to a virus that's just accumulating mutations and changes over time. Okay, so I see that a lot of us picked membrane enclosed organelles. So here we're talking about eukaryotes. So what do eukaryotes have? That's different. So eukaryotes, we have membrane enclosed organelles. That means like our nucleus has a membrane, our mitochondria has a membrane. So we have that. Bacteria and archaea don't have these. And also what eukaryotes have is DNA is found in our nucleus. For other domains of life, like bacteria and archaea, they don't even have a nucleus. They have like a nucleoli. Their chromosomes are not like, they don't look like, those. They're actually circular chromosomes. So we don't have circular chromosomes. We have membrane enclosed organelles, well, eukaryotes. We have membrane enclosed organelles, and our DNA is found in our nucleus. Actually, this red one here, presence of flagella, is not found in eukaryotes. Flagella is only found in bacteria. Okay, I'm glad that we got this right. Okay, so a nucleosome is basically just referring to a histone protein that's wrapped in DNA. So that's what a nucleosome is. We refer to it as beads on a string when we have multiple of them. So yeah, it's not nucleus containing RNA, and it's not this thing. It's basically just histone protein wrapped in DNA. Okay, so so all amino acids, I'm just gonna pull it up because it is way easier to show it. Okay, so all amino acids, no matter what amino acid we're talking about, they're all gonna have the structure. They're all gonna have an amino group. They're all gonna have a carboxyl group, and they're all gonna have an R side chain. And the R side chain is what is different. So if you just block out the R side chain, actually just don't look at it. The other thing, like what you see at the top, the amino group, the H bond, and then the carboxyl, all amino acids will have that. The only thing that's changing is the side chain. So if we look at all the amino acids here, I don't like that one. If we look at the amino acids here, so look at the highlighted, like the yellow box, the green box, and the purple and the blue boxes. Those are the R groups. Those are the things that's different. Now look at the top, look at the other, like this part, the top parts. See how they're all the same? They all have an amino group. They all have that one bonded hydrogen, and they all have a carboxyl group. So that's why the answer would be this: amino, carboxyl, and R group. Okay, so yes, the answer would be covalent. And let's talk about why. So hydrophobic means that it's water fearing. And hydrophobic amino acids, they're going to be primarily composed of carbon and hydrogen atoms, because they're like, you think of it as a lipid, you know, lipid is hydrophobic, and lipid is like hydrocarbons. So carbon and hydrogen. So if we have carbon and hydrogen, these are not capable of hydrogen bonding, because hydrogen bonding is it's only capable with hydrogen oxygen, hydrogen nitrogen, and hydrogen fluorine. So hydrogen carbon would not be capable of hydrogen bonding. So that's why it would not be the yellow. And let's talk about why it would not be ionic bonding. So only electrically charged amino acids are capable of ionic bonding. And when I say electrically charged amino acids, I'm referring to acidic amino acids and basic amino acids. And if you like look at it, acidic and basic amino acids, they are electrically charged, and you will literally see the charge on the amino acid. Okay, well, it's like, okay, let's see, I'm just trying to like show, hold on, sorry. So you see the charge, so this is basic and these are acidic. And you see the side chain, see how here there's like it's like you can barely see it all right here. Okay, you see how it's, oh my God, what happened? Well, there's a plus sign on it. So just look for those, like the electrically charged amino acids, you'll see like there's a plus or a minus. And if you see that, that means that it's electrically charged and it's capable of ionic hydrogen bonding, or ionic bonding, not hydrogen bonding, sorry. Last question. Yeah, so secondary structure. So secondary structure, there's two subtypes. There's alpha helix and there's beta sheets. These are the secondary structures. Tertiary, tertiary is basically just the secondary plus some more interactions. But here we're just looking at an alpha helix. We're not looking at any other interactions between the helices. And it wouldn't be primary, because primary is just literally just the sequence of amino acids linked together like a polypeptide chain. Okay, we're gonna review some important concepts. I've written down some notes for you guys. And we're just gonna, just review it. So two types of stem cells. We have, embryonic, which are unspecialized, undifferentiated stem cells. And we also have adult, like tissue-specific stem cells. These are also unspecialized. So we're talking about like pluripotent versus multipotent. So this would be pluripotent. They can develop into any type of cell, whereas adults tissue-specific cells, they are still unspecialized, but they are multipotent. So they're a little bit more limited than embryonic stem cells. So that's like, you should note that about the two types of stem cells. There's like embryonic and there's like adult. And then here I have written down some like different cells and what they do. Our skin cells protect our body, muscle cells contract, nerve cells send signals, and stem cells have potential to become any type of cell. So again, talking a little bit more about stem cells, we just talked about this. Pluripotent stem cells are only found in embryos, whereas multipotent are found in adults. And somatic cells, these are body cells, diploid body cells. Somatic cells refer to any type of cell except for gametes, so except egg cells and except sperm cells. Those are haploid. Somatic cells are diploid. So, okay, now I want to like host a poll for the question. So the question is, are undifferentiated cells the same as stem cells? So wait, what? Undifferentiated cells are the same as stem cells. Basically, stem cells are undifferentiated. So just remember that, like stem cells, they have the potential to become any type of cell. That's what undifferentiated means. It means like it doesn't know what it wants to do in life yet, but it can do anything. So undifferentiated cells are the same exact thing as stem cells. Okay, so now let's move on. So stem cell gene pathways. So proto-oncogenes, these are genes that encode protein that regulate growth. So when this gene becomes mutated, it becomes an oncogene. So we go from proto-onco to onco. And yeah, this is regulating growth. So it could be like cancer growth. So we can think of it as like kind of like bad if it gets out of control, because this can lead to cancerous growth. And then another type of gene is a tumor suppressor. You can think of this as like the opposite of proto-onco. It's like combating the oncogene. So since cancer cells replicate a lot, these put a brake to cell division. When turned off, it leads to cancer growth. So tumor suppressors, we want to think of them like they're good. Like they come into the rescue and they like stop the cancer growth, the opposite of a proto-oncogene. And then another good term to note is phosphorylation. This is when a phosphate group gets added. And in this case, like with this context that we're talking about, it also removes tumor suppressors. So I'm going to launch another poll. So the question is, if a proto-oncogene mutates, what will be the likely result? Is it going to slow down cancer growth? Is it going to accelerate cancer growth? Or will nothing happen? Okay, so the answer here would be it's going to accelerate cancer growth. Let's talk about why. So we know that proto-oncogenes, when they're mutated, they become oncogenes, and oncogenes accelerate cancer growth. So these are like cancer cells if it's mutated. So if a proto-oncogene mutates, it's going to become an oncogene, and an oncogene promotes rapid growth, rapid cell growth, and this is like cancerous growth. So in this case, it would be accelerates cancer growth. Okay, cell cycle. So this is the cell cycle right here. We start at G1, and we learned about mitosis in the last exam block. Mitosis is part of the cell cycle, but it's this really like small part right here. So we start at G1, growth, S, DNA synthesis, and then G2 is just some more growth. It's just getting ready for mitosis. And then, we go into mitosis. And if the cell were to go into G0, this is cardiac arrest, it's going to happen right here. Like, and it's not even going to go to G1. It's just going to go to G0. So second poll or third poll. So the question is, considering that a normal somatic cell is diploid, what would be the ploidy when it's in the S phase right here? Just end it. So yeah, the answer is C. So we know that the normal somatic cell is diploid, so it's going to be diploid right here. So now when it goes into the S phase, it synthesizes its DNA, duplicates it. It's going to go from diploid to tetraploid. 2n times 2 is 4. So that's why it would be tetraploid. We're on time, so let me just finish these slides up. Okay, so just talking a little bit more about evolution. I have like a little mnemonic here for you guys to like remember the different like mechanisms of evolution. Never forget dinosaur memories because dinosaurs are extinct. Never forget them. So the N would stand for natural selection. This is one mechanism of evolution. The mutation is memories, like that's where it comes from, mutation. And then F is going to stand for gene flow, and then the D in dinosaur is going to be genetic drift. So altogether, natural selection, mutation, gene flow, and genetic drift, these are the 4 mechanisms of evolution. And we talked about how gene flow is like movement, incorporation of alleles from one population to another, whereas genetic drift is like a disaster, a catastrophic event. And yeah, that's pretty much it for this section. And a couple hypotheses you should know. Panspermia, we talked about this, how it's a theory that life evolved from outer space, like some bacteria came from outer space to Earth and that kickstarted everything. And then RNA world hypothesis is like basically just referring to like, the earliest genomes were made of RNA. RNA started everything. So these are just a couple hypotheses that you should know. Okay. So unfortunately, we're not going to have time for the concept bubbles, but basically I just wanted us to like write down what we knew about each of these. So yeah, unfortunately, we're not going to do that, but I don't think it would have worked out anyway with the chat feature. So let's go into closing now. I really want to know where you guys are with this, and I want to know like your main takeaways. So like the main thing that stuck with you today, like pretend after the session you saw your friend and they asked you to tell them something about bio, like anything, they just want to learn about bio. What's the first thing that would come to mind? And I would love to see this in the chat. Like one takeaway, if there's one thing that just really stuck with you, what would it be? Antigenic drift and shift, that is a good one, yes. Antigenic shift versus drift. Shift is like mixed viruses, and drift is mutations. Perfect. Okay, can I see some more takeaways maybe? I would love to hear this from you guys. DNA lagging and leading strands. Yep. Lagging and leading. Panspermia. Oh, okay. Yeah, panspermia, it was like he like slipped it in. Like he didn't even talk about it for like a minute. Like I think it was just like a little like quick fact, but yeah, I'm glad that we went over it. Panspermia, outer space theory. Stem cells are undifferentiated. Oh, that's a good one. Yeah, I feel like a lot of you didn't know that. Stem cells are equals undifferentiated cells. Okay, be a few more. I think there's a few of you that haven't told me your takeaways yet. Oncogene increases rapid cancer growth. Yep. Oncogene increases cancer growth rapidly. 4 mechanisms of evolution. Yes, that mnemonic. I'll write it again. Evolution, never forget dinosaur memories. Well, yeah, that's pretty much it then for today's session. I wish you guys the best of luck on the exam. I hope today's session helped. I really just wanted to, get in some last minute review. If you have any questions up until the exam, I'm here for you guys. You can email me right here.